Okay, so here we go. Hopefully this section will be quite brief. Okay, brief meaning under 2 hours, because the muscular system is pretty straightforward. The biggest part of this lecture that you're going to have to pay close attention to is how the nervous impulse, which we did when we talked about nerves, is then translated into an actual action of the muscle. That's probably where the majority of our questions for our final exam will probably come from, or a final test. So couple of very quick questions right here. You should already know the answers to them, so we will fly through the first couple of slides very quickly. So question, which of the following types of muscles is voluntary? And you should all know that right now. It is smooth. It's skeletal muscle. Smooth and cardiac sort of go on their own. Okay. Question, which of the following types of muscles is nucleated, meaning it has a nucleus? That is skeletal. Okay, cardiac and smooth muscle do not have nucleuses in them. And the other one, which of the following muscle types is not controlled by hormones? Oh, gee, guess which one it's going to be again? Skeletal. Okay, you notice here that the ones that's controlled by that are not controlled by hormones, that are voluntary, are nucleated, that are a little bit more complex. The other ones that are not voluntary and are controlled by hormones are cardiac and smooth. Those are your involuntary ones. Okay, when we're looking at a muscle, and this is a skeletal muscle as well. Okay, and if you don't believe why it is actually skeletal muscle, take one look here. What is this attached to? Bone. If it is attached to bone, then it is going to be if it's bone, it's attached to it. If it's attached to bone, it is skeletal muscle. Okay, it's attached to your skeleton, which is a bone. It's skeletal. So the other thing to look at right here is when we have these bands of muscles, what's very interesting is they're almost like cables. Okay, when I say cables, if you know anything about bridges and stuff like that, you see those big cables that hold up the San Francisco Bridge and everything like that, and a suspension bridge. Those big cables actually have a bunch of little cables inside of them. Okay, and then they are covered by a big protective sheath. This is the same way that skeletal muscle actually works, and so you have a bunch of muscle fiber cells encased in a coating, and then you have all these little bunches as well that are encased by a bigger coating, and you have a vascular supply for each one. We see that the thing that goes actually around them. Okay, we see that a facet. I can't even speak this one today. This facet is basically covers a bunch of muscles. Okay, and then when we look a little bit later, we look at again then by this thing called perimysium. We have a circular, and then the epimysium kind of different. So this is the wrapping that goes around all of this. And then we have smooth muscle. Look at what smooth muscle looks like, and where this should actually go. Hey, wait, I've seen these little things before. Is in pathology when you were looking at when we were talking about atherosclerosis. The atherosclerosis right here. Okay, we saw that smooth muscle cell came when we started having atherosclerosis going on. Smooth muscle cell migrated from the lower levels out to the upper layer of the vasculature to then create the fibrous cap. Okay, so here are the smooth muscles. Notice they look like little worms, tapered at both ends. Okay, and they stack up on top of each other. Okay, so this is a circular layer, so we can look at them going this way, and then if we were to cut this down the middle, flip it, and then look down the center, this is what the longitudinal cut looks like of it. And notice where it is, well, you have a mucosa, then you have a submucosa, and here in the underneath the submucosa, you have the smooth muscle. And notice here as well, what's interesting is you have a set of, and this is what's interesting, you have a set of muscles. Look at how these are arranged right here. I know I said we'd slice them in half, but look at on this one right here. We have a set of muscles orientated, all going with their ends like this, going in this direction. And then we have another layer going basically perpendicular to it, okay, or almost like, if you're making a plus sign, right? You have some going in one direction, and then you cross the plus sign by putting a line in the other direction. And this is the situation that we have right here. We have some going in one direction and then some going in the opposite direction, almost on a perpendicular axis to them. Okay, and this is where when you get contractions, you can actually control, where we get peristaltic reactions, like for example our intestines that we talked about a week ago, if you had come to class. Cardiac muscle is a little bit different. We have cardiac muscle wrapping around like this, and this is very interesting in the heart, how it wraps in these directions here and then comes around like this. Now, with muscle fibers, or we talked about a muscle fiber, this is a skeletal muscle. It contains bundles of myofibrils, okay? And then the myofibrils then have something called a sarcomere. The sarcomere is split up into two pieces, a thick filament and a thin filament of a thin myofilament. Anytime you see myo, you're thinking muscle, muscle, okay? The difference between the thick myofilament and the thin myofilament is something called myosin on one and actin on the other. And this is going to be a very cool little thing about how these things sort of work. When we look at the microscopic, and if we look closely at microscopic anatomy of skeletal muscle, we see that they have this sarcolemma, which is basically a specialized plasma membrane that encircles the muscle cell. We have myofibrils, long organelles inside the muscle cell. There is a light band and a dark band, give the muscles their striped appearance. And this is what we will look at when we see it. So there's this sarcolemma, okay, so circling around all these little bands on the inside, and then we have the myofibril inside, and we see that this is where the striping, what they're talking about, this seems to be like sectional and striped all the way through. And then when you see all these stacked up inside of a band of muscle, then this is where you get these, and they all line up. You end up seeing these dark and these light bands. So the myofibrils and the banding pattern within them, as I said, there's a light band containing only thin filaments. So this is where the dark and light come in, okay? Light filaments is in the I band, okay? Which is the light band, has the thin filaments, okay? If you're thin and you don't weigh a lot, you're light. Think of it that way, okay? So the I band, or the light band, okay, Jerry says this is a good way to remember it, the I, or there's an I in light, okay, contains only thin filaments. And there is something called a Z disc that is in the midline of this, and it's sort of like an interruption. This is where you see that it looks almost like a segmented group. Okay, there's an actual disc in between the two of them. Then you also have an I band, which is a dark band. Okay, the A band, which is the dark band. Okay, this one primarily contains thick filaments. Okay, oh my God, he was so heavy and he blotted out the sun and it was dark. Whatever you want to do, or it was so big. The Godzilla was so big, it blotted out the sun and it was dark. So think of thick, big filaments. Okay, thick, big filaments will blot out the sun and make it dark. All right. So otherwise, A band, you can think of an A, and Jerry likes this. This is pretty smart of Jerry. In the first letter, okay, don't confuse with the second letter. The first letter, not the first letter. The first word in dark band, you have an A. That's the A band, thick filaments. Okay, there is an H zone which is lighter in the core, and there is an M line which is the center of what we call the H zone. And so when you line all these up, what does this mean? You have thin filaments and you have thick filaments with these sort of overlapping areas in here. We have this H zone in here. We have this Z disc right here. Okay, and this is the important part. Jerry, if I were Jerry, I would probably put this on your test. Okay, label this. If you were to be so evil, we have the I band right here. We have the A band over here, okay, and then so we have, and look at where we have here. In the I band, you have the Z disc right here. In the A band, you have that H zone, okay, and you have those overlapping pieces right here, okay, and the M line is the middle of that H zone, okay, it's the very middle line. So we have myofibril or fibril complex organisms composed of bundles of myofilaments, and look where the thick is, okay. The thick filaments that have actin, it's pointing to the little blue one right here. The thin myofilaments, I know it doesn't look like right, it doesn't look right, does it? The thin one is the thin little blue line, and the thick one over here, sorry, the thin myofilament is the little blue line, and the thick myofilament is over here. I know it's hard to see what it's actually pointing to in this slide, but the thick one is pointing to the thick line, okay, and the thin one is pointing to the little thin line. And notice very closely here before we go on to the next slide, they overlap. Okay, you have a thin filament going here, and you have a thick filament going over here. Do they completely overlap? Meaning, is this thick filament going all the way up to the end over here, and this thin filament? No, they don't completely overlap here. So there's some space for overlapping here, which is kind of important. All right, so myofibrils, we have a sarcomere. It's a contractile unit of a muscle fiber. So a sarcomere is when we say this is one functional unit of the muscle cells. You put a whole bunch of these functional units together, you get the whole muscle fiber. It's an organization of the sarcomere. We have myofilaments that produce the banding striped pattern. We have thick filaments. Okay, we have thick filaments of myosin filaments. We have thin filaments which have that actin. This will definitely come up on a test. Any test I would put on, I would have this on. The thick filaments are composed of protein myosin. Surprise that. Contains ATPase, okay, or what an ATPase enzymes. So think for a minute here. Anytime I said in this class or pathology or any other class or even pharmacology, if you had pharmacology with me, if you have something that has an A's on it, then it is an enzyme. And so this is an enzyme that, guess what it breaks up? ATP. We know that ATP is the functional unit, okay, that we need to make things move in our body. So guess what this enzyme does? Breaks up ATP so that we can release the energy that's in it. So it possesses myosin heads. So on the myosin, we have something called a myosin head. And these heads are known as cross bridges when they link thick and thin filaments during contraction. So guess what happens? You need ATP to cause a link between a portion on the thick filament and the thin filament, and this is what's going to be your pulling action that happens. The thin filament is composed of something called actin, and actin is anchored in the Z disc. So there has to be something that is these filaments have to be anchored on something so that you actually get a contraction or shortening of the muscle fiber. And where we see the major anchor is on the Z disc through the thin filaments. If you look on the previous slide, you can see that they're linked together like that. So this is what, if we look right into one of that functional groups and blow it out, okay, and look at it very closely, look what we have here. We have the Z disc, which almost looks like the thin filaments are continuous with the Z disc. And when I was learning muscular physiology, that's what I learned, is that these ones are actually, they're continuous and they're all linked together. Look at, so that means that this thin filament here is linked by the Z disc to the other thin filament over here. So there would be, if you look at this functional unit here, picture taking this, moving it over and attaching on here, and you would have another functional unit here on the other side. So a functional unit, the sarcomere, is from the Z disc to the Z disc. We have the M line in the minute. Look how these thick filaments just sort of sit in the middle here. Okay, there are these little myosin heads right over here, and here is the here's the thin filament. So this is where there's going to be an attaching that happens here through the use of ATP, okay, and then something is going to occur to actually cause a shortening where each one of these myofilaments, each one of these actin filaments gets pulled along the thick filament, okay, and then you have a little bit of space here for it to actually shorten. Now, in an actual sarcomere, the actual amount of space that is actually shortened in one sarcomere is very, 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 very small. Okay, we're talking on you know, micro measurements, all right? But when you put a whole bunch of these sarcomeres together and all of them are contracting at the same time and contracting small amounts, add up all those small amounts, and that's where you get, and that's where you get the whole contraction of the muscle. There are millions of these in one muscle fiber. Okay, 1,000,000 times a micromillimeter is going to be a bunch of millimeters. Okay, I'm saying this facetiously, meaning I'm just saying this as an example. But if you have a million sarcomeres all moving just a very micro measurement, a micro measurement altogether, the overall result of all of them shortening at the same time will give you that contraction that you see in muscles. So the microscopic anatomy of a skeletal muscle, notice that Jerry is focusing a lot on skeletal muscle because it's the most coolest. And if you learn and obviously something that is the most coolest is going to also be the most complicated. So if you learn the most complicated, then the other two, cardiac and smooth muscle, are a lot easier. So you have what's called a sarcoplasmic reticulum, or the SR. It is a specialized smooth muscle endoplasmic reticulum. That is a cool word. Try and say that 10 times fast. Stores and releases calcium. So when we're talking calcium. If you remember anything from from pharmacology, which I'm sure you all have forgotten after doing the test on the 14th, or sorry, on the 7th, you will know that anytime there's calcium involved in here, we are talking about a impulse going on through here. So stores and releases calcium and surrounds the myofibril. Stimulation and contraction of a single skeletal muscle. Guess what's going to be on this test, damn it. Okay, we're going to be talking about muscle, we're going to talk about impulses here, and we're going to be talking about skeletal muscle. Okay, so irritability, also called responsiveness, the ability to receive and respond to stimulus. So how irritable is your muscles, or how sensitive they are to actually respond to something. I like the word how sensitive are, and that matches up with patho as well. That matches up with pathology and pharmacology. How sensitive is that muscle? How ready is it to respond? We use the term irritability. Contractibility is the ability to actually shorten, and once the stimulus is received. Extensibility is the ability of the muscle to actually be stretched. Okay, and elasticity is its ability to recoil and resume its resting length after being stretched. The nerve stimulus and action potential. So yes, there's an action potential, comes down the nerve, that action potential gets transmitted to the muscle, and this is when motion actually occurs. Skeletal muscles must be stimulated by a motor neuron, like we talked about when we talked about nerves, to contract. The motor unit, or one motor neuron, okay, and all the, so a motor unit, one motor neuron, and all the skeletal muscle cells that it actually attaches to. That's a simple one. So here we have a spinal cord that we talked about before. Okay, we have an efferent neuron coming down here as well. That efferent neuron then leads up to an area, and wherever it actually connects on to those muscle cells, that is your functional unit there. Okay. The nerve stimulus and action potential. Okay. There is a junction between the nerve and the muscle. That is the neuromuscular junction. Okay, because we're changing a little bit of cellular material here, right? We're going from nerve cells that we already talked about and you already tested on. So we go from nerve cells to the actual muscle cells. So it's not a continuous, homogeneous set of cells we're talking about here. So between the two, there is a junction. The neuromuscular junction, the junction between muscle and nerve cells. It's the associated site of the axon terminal and the motor neuron and the sarcolemma of the muscle. So all those things are going to be in the neuromuscular junction. Know that. The neurotransmitter, okay, that gets transmitted from that goes across this junction is called acetylcholine. It's the thing that actually stimulates the skeletal muscle. And we know that acetylcholine in many parts of the body is an excitatory neurotransmitter for the most part. So, nerve stimulus comes down the nerve. hits neuromuscular junction, there is a gap. In that neuromuscular junction, there is a gap between the nerve and the muscle cell. Gap meaning space. Okay? The nerve and the muscle do not make contact. The acetylcholine gets released from the nerve, goes across the gap, fills the interstitial fluid, and then reacts with receptors here on the muscle. Okay, this is important for those of you who are going on to semester 3, which is most of you. Okay, when you look at this, I know my jokes are very bad this morning, are they not? The receptors over here, when we talked about that irritability, how responsive is this muscle going to be? In semester 3, when you're in pharmacology, when you're talking about drugs that cause relaxation of smooth muscle and stuff like this, and drugs that affect muscle tissue, guess what those drugs are blocking? If you block those receptors, is the ACH, the acetylcholine that is released across here, is it going to be able to do its thing and cause a contraction? No. So when you're talking about drugs next semester that deal with the inhibition of acetylcholine or blocking of a receptor on a muscle cell, ding, this is where the anatomy comes into play. So this slide. Much of this is going to be on your test. Okay, why? It's a lot of information crammed into one slide. When Jerry clams a lot of information into one slide, look out, it'll be on your test. Transmission of the nerve impulse. So when the nerve impulse reaches the axon, this is where the calcium comes in. The terminal of the motor neuron, calcium channels open, and calcium enters the axon terminal. When calcium goes into a nerve terminal, okay, so the entry causes some synaptic vessels. Calcium goes in, something's got to come out. So there's always this exchange that goes on in the body. Calcium goes in, okay, then the vesicles holding on to acetylcholine merge with the outer wall, the membrane of the outer membrane of that nerve cell, and release its acetylcholine into the synaptic gap. Okay, that's as simple as it gets. Calcium gets in there. Okay, calcium gets in. Calcium causes calcium stops. Calcium flows into that muscle cell. Okay, acetylcholine is released. You get a nice big contraction. All right. The more calcium that goes across, the more acetylcholine that's released, the more contraction that's going to happen. What happens on the muscle side once that acetylcholine, okay, actually crosses the synaptic gap? Well, sodium rushes into the cell and potassium leaves the cell, depolarizes and opens more sodium channels. Sodium in, potassium out, depolarization occurring. Once started, the action potential cannot be stopped. Contraction occurs. So you end up having a change in muscle membranes. Okay, these muscle membranes having a change in the membrane. You have a change in the membrane of the muscle cell, and then an action potential occurs, just like we talked about in nervous tissue, where once you reach a threshold, the action potential goes. Acetylcholinesterase breaks down acetylcholine and ends the muscle contraction. So there is an enzyme, that ACHE, okay, acetylcholine esterase, enzyme esterase, breaking down acetylcholine, okay, all right. Basically dissolve, once you get rid of the acetylcholine, then it stops this entire process from the very beginning. Because what did this whole process begin with? Of the muscle contraction, going up 2 steps, 1, 2, over here. We have acetylcholine in the gap. Acetylcholine causes this to occur, and then you have a muscle contraction, and then an enzyme goes back up here 2 steps, removes the acetylcholine. This returns back to a resting potential, and we're cool, and relaxation occurs. Transmission of nerve. So again, let's look again at transmission of the nerve impulse to the muscle. Cell returns to resting potential state when potassium ions diffuse out of the cell. Okay, sodium and potassium pump moves sodium and potassium ions back to their original positions, and we are now back into relaxation. Right, all of this happening in this little area in here. Okay. This diagram shows everything wonderfully. I would say probably if I were you, very quickly draw this diagram very scribbly and very cartoonish on a piece of paper and fill everything in very quickly. You'll be fine for the test of that if you can do that. Action potential reaches the axon, okay, comes down here. As it flows across the outside membrane over here, calcium channels are located here. That action potential causes those calcium channels to open. Calcium rushes in. Calcium rushes in. These vesicles that are hanging around going, hey, when do we get to leave? Okay, oops, calcium's here. They go over here. They merge to the outside wall. They release their contents into the synaptic gap. Acetylcholine diffuses across. When we say diffuse across, it happens in microseconds. Okay, diffuses across very quickly. Okay, and binds to the receptors on the sarcolemma. Okay, binds to those receptors, and then if there's enough acetylcholine there, we start an action potential on the muscle. And this is sort of I don't know how much I like this diagram, but it's a muscle. So that idea that you start a spark or you start an action potential, it then flows through the rest of the muscle like a flame starting off on the end of a very dry twig. Okay, once that flame, that spark happens, boom, the whole thing burns up very quickly and it runs across it. So the flame ignites the twig, and then the twig goes all the way across like this. So the same thing happens here. Look what happens in the muscle. You have that nerve action potential coming down to the end here, going to that terminal end that we just talked about on the previous slides. All that fun stuff that we talked about on the previous slide happens here. Once an action potential is started, it flows in this direction and it flows in this direction across the cell, across all of the cells, and all of the functional units in either direction get stimulated. So you really only need one nerve attaching to one group of muscles, and boom, it'll start off a chain reaction that basically goes in either direction. When I say either direction, picture this muscle fiber going off the end of the page and going off the end of the page on the other side. It's a huge, big, long muscle fiber, and the nerve fiber attaches dead center in the middle of it. We're saying this just for argument's sake. If we get enough stimulation and an action potential there, we will get a firing that sends an action potential along, a muscle action potential, firing off on this direction and firing off on the other direction, so that the whole muscle fiber will contract by only one unit getting stimulated. Okay, because that action potential flows from that one functional unit that received that stimulation and sends it to, in the other directions. It's a chain reaction. We get fire burning in this direction, we get fire burning in the other direction. So the muscle mechanism of muscle contraction. So once we've got that action potential, what the hell does that action potential do for us? This is going to be on your test too. So calcium binds to regulatory proteins on the thin filaments, okay? And when it does that, it exposes something called, so there's more calcium going on here. Okay, there's two points of calcium. Don't get your calciums mixed up. Calcium will flow, okay, at the neuromuscular junction to allow vesicles to be released of acetylcholine. That's spot number one in this topic. Spot number two, an action potential flowing along the muscle, length of the muscle fiber, will cause calcium to bind to thin filaments. Don't get your calciums mixed up. Now, if you think back to pharmacology, which I'm sure if you're listening to this on Saturday or Sunday this weekend, you've probably forgotten it already, but think back to those calcium channel blockers. Here we go. This is our concepts that we're working with here. So we have calcium binds to regulatory proteins on the thin filaments, exposes myosin binding sites, allows myosin heads on the thick filaments to attach. Okay, so the calcium binds to a part on the thin filament, and by doing so, it's almost like it opens a door for these thick filaments to attach to them, and they want to attach to each other. They're attracted to each other, but it's almost like there's a door in between the two of them. The attached heads then pivot. There is actually a change that happens in the thick and thin filaments, and what ends up happening is there's actually a chemical change that occurs to the structure, okay? And what ends up happening is that movement of that structure, it all occurs in one direction. And then what happens is you end up getting a contraction, a very small movement, but if enough of them together do it, the sum of all of them creates a large change in the muscle length. Okay, look up where the calcium confines and what provides the energy for the sliding process. Guess what? We already talked about this. Already mentioned it in previous slides. ATP. So when we look at this, this is where we see the difference happening. So the functional unit from Z to Z, okay, from Z disk to Z disk is our functional unit, and we have the action potential coming down, flowing across in either direction. One action potential here gets shipped to this one, and we get that change in calcium here that exposes those heads. And then what ends up happening is they lock onto each other. There is a chemical change in the actual. It actually occurs in these little heads here. What actually is these little heads actually bend over, and so they lock on, bend over. And by bending over, once they've locked onto something here, and then they bend, pulls the thin filament over top of it. So we see that the thin filament actually moves. Notice the difference here. Look at the space that's moved. The thin filament moves over top of the thick filament. That bending process pulls the thin filament in this direction here, to the point where we get up to the H zone. They do not cross the H zone. You don't see this thin filament extending over here over to the other side. Only goes up to the H zone. Yes, I know, it's only been, so the length of any functional unit, the distance that a functional unit can own, for one functional unit, the distance that it can actually contract is only the length of the H zone. That's why the H zone is important, okay? And because you've got thin filaments moving this way, to the end of the H zone, or to the middle of the H zone. You have a thin filament moving this way. Okay, the sum of a filament moving this way and the sum of a filament moving this way is the distance between them. So that is the functional length that they can actually contract. Notice that everything else stays the freaking same. The I band is the same length. The Z band doesn't move. Everything, it all happens in here, and it's an overlapping on either side of the M line. And this is what I was talking about. What actually happens when we get this linking going on? So let's look at what happens here. We have a protein complex that gets exposed by calcium, right? And then we end up getting a locking that occurs here. Okay? Here we have it happening again, where we see this, and there's a little locking that occurs, and then we say the upper part, and we see that there will actually be a little bit of a movement that occurs from this. Here's what I was talking about. I know I skipped over the last two slides. So calcium is exposed. We have the link up between the two. Notice what happens here. When these link up, as I said, the heads on these thick filaments will chemically. This is a chemical. This is a chemical property right here that's occurring. What's happening is once it links up, there's a change in the molecular structure of this head. So much so that it almost bends over. It's like I'm trying to think of something here that you would probably have in your. Oh, okay. Go get it. If you have any food left in your house right now, go get a banana. When you peel a banana, you start at the top. You break the top, right, and you bend over the outside of the banana peel, right, and the banana peel bend and you pull it. Go it from top to bottom, you'll break the top of the banana, pull the banana all the way to the bottom, and guess what? The outer strip has moved down to the bottom, hasn't it? And if you're really anal retentive and go, oh my God, I don't want to eat this banana right now, you can reapply that peel back up to that banana and cover the banana on the inside, right? Conceptually, that's what is happening. I'm not saying this is a banana, but I'm saying conceptually what's happening is that this piece right here is actually, there's a bending process that's happening, like pulling the peel of the banana all the way down to one side. That happens, so you're actually contorting it, and then when it relaxes, it goes back, like the banana peel going back up to where, putting it back together. And by doing that, that's not a long distance. The banana is not a huge thing, but when we're talking about like thousands and millions of these doing this all at the same time, locked onto a thin filament, you can see where the thin filament will move because it's already locked in. So with the banana analogy, you have a thin filament attached to the top of the banana peel, and then as you peel off the banana and move that banana peel down, okay, the thin filament will move. Okay. So there's a video for this. I'll make sure that this video is also posted as well for you, and I think you should probably watch that as well to conceptually get an idea of this. When I was in university, it was a hard concept for me to get as well. And the contraction of the skeletal muscle, the muscle fiber contraction, it's an all or none sort of thing. You either have enough stimulation to do it and it contracts, or it doesn't. Okay, there's no little contraction that happens. Okay. With a skeletal fiber, it's not all fiber. So, but you're probably wondering, but hey, how do I get a stronger contraction? I went to the weight room this morning. How do I get a stronger contraction? Well, it's simple. Don't contract all the muscle fibers. Remember, you have muscle fibers, like that cable thing. You have muscle fibers contained in an insulating membrane, right? What if you only fire off some of those fibers? Remember, there's a nerve going to each muscle fiber. So how many muscle fibers you recruit? You don't have to recruit them all. So to have a gentle contraction that has less force in that cable, okay, where all those muscle fibers are, if you only activate 10% of those muscle fibers, right, it's an all or nothing principle along the muscle fiber. And the muscle fiber is inside, okay, that analogy I gave of a bunch of fibers inside of a cable. If you only fire off a few, or say 10% of those fibers, then you're only going to get a 10% contraction of what you're able to do. If you fire them all off, then you get an Arnold Schwarzenegger sort of bodybuilding oomph going on. If you fire all of those fibers, then you're going to get the maximum contraction that you can get out of that muscle group. Okay? So within the muscle fiber, not all fibers may be stimulated during that interval. Different combinations of muscle fiber contractions give different responses. And as we say within the term we use is there's a graded responses, different degrees of skeletal shortening, depending on how many skeletal muscle fibers you have activated. So muscle response to a strong stimuli. Muscle force depends on the number of fibers stimulated. It's just sort of elucidated that to the previous slide. Contraction of more muscle fibers results in a greater muscle tension, and muscles can continue to contract unless they run out of energy. What's the energy? ATP. ATP. So ATP is the immediate source of energy for muscle contraction, stored in the muscle fibers and in small amounts are quickly used up. After its initial time, other pathways have to be utilized to produce ATP, so anaerobic versus aerobic respiration. 3 ways to generate ATP. Direct phosphorylation of ADP by creatinine phosphate, which is aerobic respiration. Anaerobic respiration is aerobic glycolysis and lactic acid formation. Okay, when we talked about we talked a little bit about metabolism and stuff like this, this is why you need air. You need air to go through the proper burning of carbohydrates so that you can turn ADP into ATP. If you don't have enough air, you start burning other stuff, and you use a different metabolic pathway, which unfortunately creates lactic acid. There's direct phosphorylation of ATP by creatinine phosphate. Okay, muscle cells store creatinine phosphate in high energy. It's a high energy molecule. After ATP is depleted, ADP remains, and you have to recharge it. Creatinine phosphate then transfers a phosphate group to ADP to regenerate, and the creatinine phosphate supplies are exhausted in less than 15 seconds. So about 1 ATP is created per CP molecule. This is the fastest way to regenerate that. So when you're actually working around and doing aerobic respiration and working on your cardiovascular system, what you're actually doing is you're not actually working on the muscles. And think about this for a second. Why is somebody who does for example, think about this, for example. Somebody who is a marathon person, like a marathon runner or the crazy crap I do with canoeing, or anybody who does anything that's a marathon endurance sport. Are these people like huge muscular people, like a bodybuilder, like Arnold Schwarzenegger? No, they are not. They do not look that big. And I can guarantee you, I have seen. It was very interesting one year when I was doing my canoe marathon that goes on for 8 to 10 hours, all at once. Start at 6 a.m. in the morning, finish at like 4 in the afternoon, no stopping. The There's a guy one year who was out of the military who's trying it for the first time. And this guy was like, huge bodybuilder sort of guy, right? This guy almost died at the end of this. My buddy and I got up and went, whew, that was a lot. Had a drink of water. Okay, let's go. Yes, our bums hurt. Let's go on. But we were like, okay, we probably could have kept paddling for another 2-3 hours if we had to. Okay? He looked like he was about to collapse and die at the end. Big muscular bodybuilder. The point I'm getting to here is building up of more muscle does not relate to how efficient your body can regenerate ATP. For my buddy and I, who paddle constantly for hours and do marathon sort of stuff, we're not huge and muscular as he was, but what our body is able to do is it's able to keep going and going and going and going without a big buildup of lactic acid. As long as I have sugar somewhere in my body, I can regenerate ADP to ATP. Think of marathon runners. If anybody watched the Scotiabank Marathon and stuff like that, or the Waterfront Marathon, those people who win it are not huge and muscular, but they can go on forever. What their body is trained to do, and what they've done, is they have efficient mechanisms in their cells, specifically the mitochondria within their cells, that can do this. Okay, that can regenerate ADP into ATP. As long as they have sugar in their body, they will efficiently regenerate ADP to ADP. So when you do cardiovascular workout, what you're working on is your body's ability to go for long periods of time and efficiently change ADP to ATP. Okay, a bodybuilder, that's different. You're actually creating more muscle tissue. It doesn't mean it's efficient muscle tissue. It just means it's bigger. All right. So direct phosphorylation. Now they've gone off of that. We have an ADP, which is the end product of using up ATP in the body. Creatine phosphate actually helps to it gives up a phosphate, ADP, adenosine diphosphate, takes a phosphate from creatine phosphate, okay, and you end up getting creatinine, which is the end byproduct, and that phosphate goes to ADP, and you have ATP, adenosine triphosphate. There we go. So basically you get a phosphate taken away from creatine phosphate, given to an ADP, you're recharged, and you now have ATP is ready to go again. So this is what I was talking about, aerobic respiration. Aerobic respiration is glucose is broken down into carbon dioxide and water. You only get that water by taking in oxygen and using oxygen efficiently. Okay, we take the oxygen, we apply it to an H, which is the end product of aerobic respiration, and you end up creating water. And then carbon dioxide is created as well. So glucose is broken down into carbon dioxide. So when you breathe off carbon dioxide, you're breathing off byproduct of respiration of a sugar molecule. Okay? So yes, you can say after eating a big chocolate bar going, look, I've just breathed off my sugar. All right? So a glucose molecule would produce at least 32 ATP. So our bodies, I don't care what you say, our bodies were built to burn glucose. All right? A series of metabolic pathways occurs in the mitochondria. So all this occurs in the mitochondria. This is a slower reaction that requires continuous oxygen. So yes, somebody who's doing a marathon and doing stuff like that, if they've built up their aerobic respiration, they've built up their cardiovascular fitness, what they are doing is they're making this much more efficient. Okay, so you have the creatine phosphate, which boom, and this is why bodybuilders take creatine phosphate, right? They're actually loading up the short the short-term method of replacing ATP. The last slide said, I'm just going to clarify this for a second, the last slide said there's creatine phosphate in your muscle cells. So very quickly, so that you can do a bunch of contractions very quickly, you will use up all the creatine phosphate. Okay, it's in the cells, and it gets used up in about 15 seconds. After that, okay, then you have to use aerobic respiration to then replace ATP within the body. This is the crap that marathon people do. Okay, we make this part in our body, and the previous example, why I was saying the example between a marathoner and somebody who's a bodybuilder, is that a bodybuilder will use up all their creatine phosphate very quickly. Their aerobic backup system, which is slower, okay, may not be as efficient. So when you do cardiovascular fitness, you are working on this method here to make it more efficient, which means you can keep going and keep going and keep going. Okay, a bodybuilder probably has a lot of creatine phosphate, but can only move a large weight for a short period of time. Why? Because it burns up all the creatine phosphate. Okay. Make sure that you remember the steps of cellular respiration. Make sure that you know what the end products of cellular respiration are, and those were in previous lectures. Okay, so here we go. The aerobic pathway, aerobic cellular respiration, breakdown of glucose to pyruvic acid. Pyruvic acid goes into the mitochondria. From the mitochondria, we do all the stuff that we talked about a couple of lectures ago, and we end up getting CO2 and H2O, and our end product is ATP. This part over here, if we can't get pyruvic acid from glucose, we will break down fatty acids, and we will try and break down amino acids. These are the other two pathways that feed into this aerobic respiration. And what this X means right here is, if I have glucose and I create pyruvic acid and I have oxygen, then what I will do is I will go through the glucose breakdown of pyruvic acid all the way through to the point where I will get my byproducts here, CO2, H2O, and ATP. If I do not have oxygen, I do not go through this breakdown. I then have to take products from fatty acid breakdown and products from amino acid, and then what ends up happening is I end up breaking those down to try and get some ATP and byproducts. The only problem is I end up getting some nasty byproducts. If we're breaking down solely fatty acids, we get ketones. If we're breaking down amino acids, we end up getting buildup of ammonia and nitrogenous products. So we can break these down. It's not optimal. You don't get the same number of ATP if you break down fatty acids and amino acids than you would from breaking down one glucose molecule. Okay? So this is where we talk about diabetic ketoacidosis. Guess what happens in diabetic ketoacidosis? For your pathology, here's a hint for pathology. In diabetic ketoacidosis, this pathway stops. Why? Because you don't have any insulin to do this first step. So if you can't do this, the first thing the body does is, holy crap, there's no sugar anywhere. Let's start breaking down fatty acids. Okay, let's start breaking them down very quickly. We have no other glucose stores anywhere in our body. So that's what happens, and then we start breaking down amino acids, and this is where diabetic slips very quickly into diabetic ketoacidosis. There is a concert going on in your body right now where the majority of it is glucose. You will get some breakdown and stuff happening through amino acids and fatty acids. This is where somebody goes on a diet and reduces their glucose intake and exercises more and everything, and you throw that balance off of it. Yes, you will start burning some fatty acids. Okay, this is where a diet where you have more output that's needed and you have less intake of food and calories. What's going to happen? Well, your whole body isn't going to completely burn fatty acids and amino acids. It is still going to burn some of the sugar that you put into it, but it might not have enough. So you start burning fat cells. This is how a diet and exercise works. Okay. Does it mean that your entire body flipped over to only burning fatty acids all at once? No, because you have insulin, you have stores, you took in some carbohydrates during the day, you just didn't take as much in as you usually did. You had a half a pizza instead of eating the whole pizza. Instead of going just for a walk, you went for a run. Okay, so in a diet where you're increasing when you're dieting, you're increasing exercise and decreasing the amount of calories that you're intaking. You have a concert going on where you're breaking down glucose, and at times you're breaking down this as well, and that's how you lose weight and you lose fat. And diabetic ketoacidosis, everything stops. There is no use of glucose once the insulin is gone. So the entire body, all at once, relatively speaking, I'm speaking metaphorically here, but the whole entire body all of a sudden switches over and says, there is no glucose anywhere in the body. I can't use any of the stores. I can't use anything. It starts burning all fatty acid everywhere all the time as soon as the insulin is gone. And that's where you get the ketones for diabetic ketoacidosis. Okay? That was your little hint towards pathology next week. All right, aerobic glycolysis and lactic acid formation, the reaction that breaks down without oxygen. Okay, so we run out of oxygen, we still have to start burning that, that other stuff. Glucose is broken down into pyruvic acid to produce 2 ATP right off the top. Pyruvic acid is then, if we don't have oxygen to take that pyruvic acid through the entire process, the pyruvic acid is converted into lactic acid. Go back to the previous slide. Notice that we break down glucose into pyruvic acid. That gives us 2 ATP right away. And then that pyruvic acid has to go into the mitochondria. If there's no oxygen, it does not go into the mitochondria and go through the entire process. You have to do something with that pyruvic acid. What do you do with it? Well, we do one thing to it. We convert it to, we want to get a little bit of an energy out of it. Okay, we didn't get all the energy out of it that we need. So we convert it to lactic acid and then pee it out. That lactic acid, that conversion gives us another 2 ATP. Okay, and then so when we have aerobic respiration, we only produce 4 ATP in total. We don't get the big bang for a buck. We don't get the big 32 plus that we get down at the bottom when we did aerobic. All right, so this is where you get lactic acid buildup. Okay, you slipped into anaerobic glycolysis and lactic acid formation, and you get very little glucose. You get very little ATP out of this, and this is where you end up getting muscle fatigue. And this is what we looked at on that previous slide, the 2 slides before, where you had, now this is exactly the same as that previous slide, 2 slides before. It's just only the top little half right here. You just don't have the little mitochondria and the other 2 pathways that I showed you here before. This is exactly the same as top. Glucose breaks down through the glycolysis. We take the glucose molecule, split it into pyruvic acid. Doing so, we get 2 ATP. We don't have any oxygen to send this into the mitochondria. Therefore, we have to do something with this pyruvic acid. We've created it. Now we got to do something with it. Can we get anything else out of it without oxygen? Yeah, we can convert it into lactic acid. We get 2 more ATP, done, finished, off we go. Okay? So without this, we only get 2 ATP. We convert it into we turn it into lactic acid, and then your body needs to get rid of that. And the duration of this, you can go, is about 40 seconds. Not very much. So if you're doing something, you go to the gym and go, oh, I'm going to work really hard, and you get 15 seconds for your creatine phosphate. You can get another 40 seconds if you do this through the aerobic pathway. Pretty much, what does that give you? That gives you less than a minute, okay? That's 55 seconds of work that you can do without any oxygen being used. Muscle fatigue and oxygen deficit. If the muscle activity is strenuous, prolonged muscle fatigue occurs because of ionic imbalances that occur because of those nasty byproducts that we're making, like lactic acid. Lactic acid accumulates in the muscle. Any ATP supply is diminished or depleted, okay? And basically we have, after the exercise, the oxygen deficit is repaid by rapid, deep breathing. So what happens is, what that oxygen deficit, meaning that you did not provide enough oxygen during that activity to go through aerobic respiration. So you become an oxygen deficit. This is where you get winded, okay? In the summer, when I do 2 sports, marathon canoeing and dragon boat racing, the dragon boat racing that I do, we only paddle for extreme exertion for about 2 minutes. In the marathon paddling, we're paddling at medium to high exertion for hours at a time. So I go from marathoning and then I jump in the dragon boat, and our coach pushes through like 5 minutes of extreme paddling. And at the end of it, my buddy and I look at each other, and probably in about 5 or 6 breaths, our respiration rates have come down and our heart rates have come back to normal. Everybody else in the boat is puking for the next 2 minutes. Okay. And she has to give them a 2 minute to 5 minute break. The two of us are sitting going, yeah, we're done, we're good. Okay. So that's what's meant by the oxygen deficit. If your body isn't trained to work efficiently in aerobic respiration, then what happens is it takes longer for you to overcome that oxygen deficit that you needed, okay, to go through aerobic respiration. Okay, isotonic contractions. This is something a little bit funky doodle. Isotonic contractions. Myofilaments are able to slide past each other during a contraction. The example, so an isotonic contraction, example, muscle shortens, and we end up having muscle movement occur. So isotonic, we have shortening of the muscle, okay, bending of the knee, and all of you can do this right now. Okay, keep your elbow straight, but tense up your muscles. All right. This is what the next thing is called, an isometric contraction. Isotonic means that the muscle actually shortens and a movement occurs. Isometric is when you just tense up your muscle, but the joint doesn't move. Okay? Isometric is basically causing tension in the muscle. The muscle is unable to shorten or produce movement. Isometric, iso, the same length. Metric system, okay? So isometric, iso meaning same length. That means no muscle movement, but there's tension within the muscle. Isotonic is the opposite of that. There will be an actual contraction of the muscle. So muscle tone keeps muscles healthy, ready to react. The result of a staggered series of nerve impulses delivered to different cells within different muscles gives us our movement. If a nerve supply is destroyed, muscles go flaccid. Okay, they then become paralyzed, and then they will atrophy, and then they will go into contractions. This is where some of your patients who have CP, or for example, cervical vertebrae injuries, somebody who is a quadriplegic or a paraplegic, their muscles are fine. It's the nerves innervating those muscles. If those muscles don't move, they will start to atrophy and they will start to contract. Okay, because they are not being used. Okay, and here's my two examples between my two dudes here. Hey, look, you see the big muscle bodybuilder right here? Look how big his muscles are. Look how thin his muscles are. So this guy is able to do a lot, is able to move big weights. Okay, but he's not able to probably go for long distances. This guy would get crushed by this weight, because look at the size of his muscles, but he could keep going on and on and on and on and on forever. This guy will outrun this guy. If you can get away from him within the first, this guy has about what did we say? About 55 seconds worth of energy. So if this guy can catch this guy in 55 seconds, he'll kill him. If this guy can get away from him within a minute, he can run away from him forever. This guy will get winded and fall over. And probably because of the steroids that he had, he'll probably have a heart attack. Okay, so this question right here, I already spoke about it on the previous slide when I was talking about the two guys. Okay, if you lose, just to recap very quickly, if you lose innervation or muscle or nerve supply to a muscle, it will over time start to contract on its own, and it will atrophy, shorten, contract. You will get contractures, and unfortunately, it will become non-functional as well. And that is it. Good luck. We did this in just 1 hour, so hopefully, I know I spoke very fast, but it's on YouTube. You can rewind and go backwards and forwards. Good luck on your tests on Monday, and good luck on anatomy when I see you on Tuesday.